I'll share. And then okay.
Yes, sir. Screen is visible.
Okay.
It is visible, sir.
Okay. I'm gonna get my pointer as usual. And okay. And I'm also going to check whether screen is not cut or anything. So just help me out. I'm taking the pointer way to the top of my slide. Can you see my pointer?
Sir, it is not being cutted, sir. It
Yes,
is
sir.
full
Okay.
screen.
Okay. So you can see my pointer at the top,
Yes, sir.
Yes,
and
sir.
now the
Yes, sir.
pointer is at the bottom.
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay. Great. Great. Okay. So we have been talking about recombinant DNA technology, and then after we learned a little bit about what is classically called as cut and paste technology, where you learned how to take uh, plasmid vectors and cut out a piece of DNA, put it in the vector, and then clone it in a sense. We then learned a little bit about another technique for amplification, which is called as PCR. And then we also talked a little bit about uh, uh, okay. Somebody's mic is on. Can you just turn it off, please? Thank you. Thank you. Okay. And then last time we talked about reflip. Okay. And amplification, fragment length, polymorphism. And and taking from there, while we were doing that, we also learned a little bit about electrophoresis. We learned a little bit about blotting, and we also learned, in a sense, little about DNA fingerprinting or DNA profiling. Okay. Again, basically using the same methodology, that is use of basically restriction enzymes, cut and paste technology, and also PCR. Another application of these methods is what is referred to as construction of uh, genome. Uh, Somebody's mic is on. Just if you can mute it, please. Thank you. Thank you. Okay. And as I said, one of the other technologies that that finds application for several purposes is what is called as construction of a genomic DNA library, or what are called as cDNA libraries, or C stands for complementary DNA libraries. Okay. Now, just to introduce you to what these things are, uh, DNA libraries are basically collections of DNA sequences. Okay. Now, we have a library in our college also. Okay. So, if you were to define our DNA book library, as we call it, uh, we will call it as a collection of different books. Okay. So, in that regard, you might think that a DNA library is collection of actually DNA sequences obtained from different species like plants and bacteria and fungi, and this set of these sequences of all the different animals constitute a DNA library. Okay, but that is not the case. Okay, if you look at the definition, a DNA library is a collection of clones of DNA designed so that there is a high probability of finding any particular piece of the source DNA in the collection. Okay, so the idea is somewhat like this. It's like saying that in a way, what I have is that in my library, I have lot of these books in which there is one page from Remington in each of the books. Okay, and you have all seen Remington, no? It's a big fat book. It has got about 4,000 pages. And imagine what I have done is taken the Remington, torn out each page of it, made it kind of separate, and then stuck this page in another book. Okay, so essentially what this other book is a book which has got everything identical except this page being different. Okay, so imagine for a moment that I buy lot of blank notebooks uh, of the same size as a Remington ka page, and what I do is I tear out Remington ka page and very systematically one page I insert in this blank notebook. And all these blank notebooks are then kept in the library. And so what you would have is actually a collection of what we call as clones of DNA. Okay, clones of DNA would be basically copies of lot of DNA. But imagine for a second that you have, let us say, Remington had 2000 pages. So what you have is 2000 notebooks, each containing one page. Okay. So essentially, all of these 2000 notebooks that I have are all containing, collectively speaking, the entire information that is there in Remington. Okay. So now what you do is this. What you do is each of those 2000 books that you have, no, you take it to our Kalpana fellow and tell him कि ये पूरा इसका मेरे को xerox करके दो और 10-10 कॉपी चाहिए मुझे. So now what you will have is if you were to look at your library, 11 notebooks, all of which contain one page of Remington, and since they are copies of the original, 10 copies of the original, they can be called as clones of that page. Okay. Then if you go further down the library, again you will find 11 notebooks. They are also clones of each other and they contain maybe a different page from Remington. Okay, so like that, if our library was filled with, actually speaking now, 2000 into 10, okay, 20,000 plus 2000, 22,000 notebooks, each 10 of them, or sorry, 11 of them, all containing the same page, you would have had what is called as the library of Remington, okay, where Remington is now taking the place of DNA. So now see the way I read. A Remington library is a collection of all the clones of Remington ka pages designed so as to be a high probability of finding any particular page of Remington in that collection. Okay, so now technically speaking, I have the entire Remington except it is not together. Okay, and what I've done is I've made clones of each page, but the entire information of Remington is still there. Okay, so something like that is referred to as a DNA library, and the reason for doing it somewhat like this will be obvious in a little while. Okay. Now, what is the function of the library? Okay, just like our library, DNA libraries are used to collect and store information. Now, what you have done is you have collected and stored information about the entire genome, okay, or the entire complementary DNA, content. Okay, I'll come to that a little later. As a library. Okay. Now, the way you store it is rather than storing is at a set of books which you find in a regular library. Here in the DNA library, everything is stored as a set of DNA molecules. All DNA libraries are a collection basically of DNA fragments that represent a particular biological system. Okay. So, if I'm interested in humans, so what I would say is I have a human genome ka DNA library. What that would mean is I have all of the genome of the human in bits and pieces put in different places in this library. Collectively, that library will represent the human genome. Okay. Now, why do we do this? Okay, and as we go along, you'll find that it will be obvious why we do this. There are two main reasons why we do it. One is so that later on we can get sequence information of all those pieces of DNA which are part of the library, and then as an extension, if I put together all these sequences, I'll get the sequence of the entire genome of whatever the organism is, say human or E. coli. And the second reason is that I can take any such clone and keep on growing it again, so I can amplify any piece I want, and that would be called as gene cloning. Okay. Now, there are two types of libraries which people usually work with. Okay. A genomic library is a library that represents fragments that are correlated to the entire genome of an organism. Okay, so you will say E. coli genomic library, Staph aureus genomic library, HIV genomic library, uh, you know, earthworm genomic library, butterfly genomic library, human genomic library, so on and so forth. Okay, so what is done in this type of library is whatever is the genome of the original organism, that is cut into small pieces. Okay, and each of those pieces is put in a vector. Okay, so here I am showing that vector, and this vector contains a small piece, and this is a part of the entire genome of a given organism. So let us say, for example, I am talking E. coli, and E. coli has about 4 million base pairs. Okay, so I cut down this E. coli of 4 million base pairs into short pieces, okay, which are manageable, and each piece I put in a vector. Okay. Now this collection of these vectors that collectively represent the entire E. coli genome would be called as a E. coli genomic library. Okay. So let us say for example, if I took the E. coli 4 million base pair genome and I cut it into let us say 50,000 pieces. Okay. Essentially now I'll have 50,000 different recombinant vectors, such that each vector would have this part same, but the insert being different. And each insert, because it is part of that collection of that 50,000 pieces, then the entire recombinant collection that I have of 50,000 recombinants will represent the entire E. coli genome. Okay. Such a library is referred to as a genomic library. As opposed to that, there is also something called as a complementary DNA library. Now, the way complementary DNA library is made is what I do is that I take a cell, okay, or more correctly, I should say a group of cells from a given tissue, okay, and what I do is break open the cells of the tissue. Let us say for argument, I am talking liver cells, okay. So I take a piece of liver, take out those cells from the liver, and what I do is I collect all the mRNA from it. And what do mRNAs represent? They actually represent those genes which are being actively transcribed in the liver, okay. Now, what this would basically represent indirectly is what all proteins that I may find in the liver, because mostly this mRNA later on will get converted to the protein. And then what I do is I take that mRNA and using an enzyme which can read mRNA as a template and make DNA, I make what is called as complementary DNA, or sometimes C is also called as copy DNA. So what I have done is, let us say I have a length of mRNA, I use reverse transcriptase, correct? Remember that enzyme, reverse transcriptase, it reads RNA as a template and makes DNA for you. So I use that information, use that ability of reverse transcriptase to convert whatever is the sequence of mRNA to a corresponding complementary sequence of DNA. Okay, and that is referred to as a complementary DNA. Now, all the different mRNAs which are present in a given cell, in this case a liver cell, will all give you different copy DNAs. And the collection of these complementary DNAs or copy DNAs is referred to as a cDNA library. So, basically, what I am trying to tell you is a genomic library is a collection of all clones comprising all the DNA within a donor cell, and a cDNA library is a collection of all clones that comprise the information that you have obtained from the mRNA within a donor cell. So if I were to ask you a question, which library would be a bigger library, genomic or cDNA? What would your answer be?
Genomic.
Genomic. Genomic.
Why would you say
So the
genomic library would
genomic
be bigger?
library would have all the genome, matlab the whole genome of an organism. So obviously it would be more as compared to the proteins.
Sir, also in the
Uh,
processing of mRNA, introns are removed, so the size decreases
okay.
as compared
See,
to
there are
genome.
so there are two reasons why cDNA library will be smaller. One, as somebody answered, especially in eukaryotes, you have got introns which are removed and all those things. And the second thing you have to remember is every cell in our body doesn't use all the information that is there in your genomic DNA. Okay, just to give you a simple example, if I were to ask you where insulin is produced in your body, what would your answer be?
Beta cells,
Beta cells of
islets of
pancreas.
pancreas.
Beta cells of pancreas. Now, is the DNA that is present in the beta cell of pancreas different from the DNA which is present, say, in your heart, in your brain, in your muscle?
No, sir.
It is
No,
the
sir.
same DNA, right?
No,
But
sir.
what is happening? Yeah. So what is happening is different tissues are expressing different genes depending on what is the functionality of the tissue. So that's why a given tissue ka mRNA will not be all the possible genes that can be expressed. Okay, and further there are a lot of DNA sequences which are junk DNA which never get expressed. So you will never get mRNA out of it, and therefore the cDNA library will always be smaller than a genomic library in terms of the size of DNA that it is representing. Okay. Now, going further, okay. So as I said, a genomic library is made from total nuclear DNA of an organism. Okay. And this DNA is cut into clonable size pieces as randomly as possible. Okay. What do you mean clonable size pieces? If you remember when we talked about different vectors, we said different vectors are able to carry different lengths of DNA comfortably. Anything more than that, then it causes the problems with respect to the functioning of the vector. So what you need to do is, since you are going to put this piece of DNA into, say, a plasmid or a yak, and then the organism which contains a given recombinant, you are going to amplify, let it grow so that you have all clones containing the same piece of DNA, and like that you have many clones. You have to cut the size so that it is comfortably carried by the vector. Okay, so in a sense it is shown somewhat like this. Okay, so let us say I isolate the DNA from cells. Okay, that is shown here. So this is the DNA which I have obtained from a given cell. Okay, now remember, because I'm talking of a genomic library and I'm going to take the entire DNA, I can pick any cell of that organism which is containing the DNA. I will get the same information from it, because every cell in a human, as far as we are concerned, all our cells contain the same complement of DNA. Then what I do is I cut this with restriction enzymes, okay, and I'll get small pieces. And I've shown 3 here, but you will actually get thousands of pieces, each being different, because there are so many different restriction sites just randomly occurring that if you treat your big DNA with a restriction enzyme, you'll get a lot of pieces. Okay. Then what you do is you take a vector and cut it with the same restriction enzyme. So you'll get nice sticky ends, right? And then you randomly allow different pieces of DNA to go into different plasmid molecules to get your recombinants. And then you take this recombinants and introduce that into a bacteria, and just like before, you test this bacteria for containing the recombinant by replica plating or blue and white colonies. And then what you do is pick each individual bacterium containing one type of recombinant and grow them separately. Now here, all the bacteria, let us say, were derived from this one, so they all represent one piece. Then let us say this one, which was derived from taking up this plasmid, when that grows in this culture, each of those cells will contain the plasmid which has the same information as this, which probably was derived from this piece. So as I look at all these different amplified test tubes, each containing a lot of cells, they will collectively represent one set which has been amplified, but together they will represent the entire genome. Okay, and such a library is referred to as a genomic library. Okay, so again, going in a little bit more detail, let us presume that this is your genome, okay? And I'm showing this entire genome as this big black line. And let us say that there are 1, 2, 3, 4, 5, and 6 different restriction sites for a given enzyme. So if I were to take this genomic DNA and treat it with this restriction enzyme, I'm expecting to see how many pieces?
7.
So remember the rule?
7.
7 pieces.
N plus 1.
N plus 1 pieces. So 6 restriction sites, I get 7 pieces. Okay. But now look at this interesting thing. You will get only 7 different types of pieces if you were to take this genomic DNA and expose it to the restriction enzyme till there is no more restriction site left for it to be cut. Okay, that means you are going to completely digest your genomic DNA. Okay, and if you do that, you'll end up getting a set of restriction pieces, but collectively there will be of 7 different types. Okay. Now, when I make the library, I cheat. I do not allow the restriction enzymes to totally cut my DNA. Okay. In other words, what I do is what is called as a partial digestion. Okay. Now, let's see what happens when I do a partial digestion. When I do a partial digestion, rather than getting 7 different pieces, I get a whole slew of pieces of different sizes. Okay. For example, look at this piece. This piece is obtained by a cut occurring here and a cut occurring here, but there is no cut occurring here. Look at this piece. This is representing this piece without a cut occurring here. This is representing this piece without a cut occurring here. This is representing a piece where the cut occurred here. Okay, so essentially what you'll have is a whole collection of pieces of different sizes. Within them, there will be the 7 pieces, but there will be also pieces which are longer. Okay, look at this one. Here, this is representing a piece which was cut here and here, but was not cut here and here. So essentially, see what you are doing when you do a partial digestion. You will find that this information, let us say sequence information here, is going to be present in those bacteria which carry this as part of their recombinant plasmid. It is also going to be present in those bacteria which contain this piece as part of their recombinant plasmid. It is also going to be present in those bacteria which carry this as a piece of their recombinant plasmid, and that information is also going to be present here as a part of this plasmid which carries this bacteria which carries this recombinant plasmid. So essentially what you are doing is making sure that this information is represented at many different places. So they will act kind of as a verification. Okay, so you'll find that when you clone the bacteria which contains this piece, get a lot of copies and do the sequencing, you'll get a certain sequence. But then that sequence will also be found here, okay, as the end of this one. It will also be found here as a middle of this one. It will also be found here at the front of this one. So essentially it acts as a check that the sequence is a real sequence. It does exist, but then because of doing partial digestion, what is happening is you are also able to do sequencing and correctly order this entire genome by use of this overlap. Okay, and that is why when you make a genomic library, you never do what is called as full digestion. You also always do partial digestion. Okay, now here is what I just said shown to you. If I do full digestion, then I'll get pieces which are non-overlapping, right? Now each piece is going to be put into a what you call plasmid, and that plasmid will be put in a bacterium. So when you are growing this, each of these bacteria clones or colonies are going to represent all bacteria which contain the same insert. Let us say, for example, here all the bacteria which are growing are representing the insert which is obtained from here, which is in the plasmid. Let us say this one is representing this one. Let us say this one is representing this one. So when I grow each of these bacteria, take out the recombinant plasmid and sequence, say, this piece of DNA, I'll get the sequence information of what was there. But then when I try to order the information, I will not know which piece comes there, where, right? Because I've done what is called as the full digestion. Now see what happens when I do partial digestion. When I do partial digestion, what happens? I have pieces which are like this, then like that, then like that, then like that. Then what happens is, when I grow each of those clones, now see, this set of sequences are represented by 3 colonies, okay? And when I clone, I'll be able to actually overlap each other and build up the entire sequence, okay? So, so this is almost like saying that if I, imagine that 10 of you are sitting in the first row in the class, and I want to represent you as a library, okay, so that I tomorrow know how you are organized in the front row. I can do that in 2 ways. I can take individual photographs of each person as they sit in the chair, okay, and if I do that, I will get information about all 10 of you, but I will never know in what order you sat. So what I do is something very interesting. Every time I take a picture of you, I make sure I take a picture of 3 people at one time. Now, I will have a multitude of pictures, each representing 3 people. But the beautiful thing is, if I look at all the pictures, I can quickly figure out what are the arrangement from left to right, correct? Because each picture is allowing me to figure out who is to the right and left of somebody. Then in the third picture, 2 will appear and a new person will appear. If the new person is on the left, then I know who was there to the left of these 3. Okay, then I take another picture where more people are together. And basically what I will be able to do is figure out who are the 10 people exactly sitting, because I took each photograph which contained 3 faces. Okay, ये बोलते हैं ना आजकल का वो कैमरा में वो एक फंक्शन रहता है where if you want to catch a wide scenery, no, the camera tells you just take 3 photographs, okay, and then in the back end, no, there is a software which sees which are the common things between the photograph, correctly aligns it, so you get a one beautiful big picture, okay. Many of you must have had that type of camera where you take
Panorama, sir.
panorama. So panorama का जो philosophy है ना, exactly वही philosophy is used here. So when panorama occurs, you have to be very careful that as you pan and take photographs, you don't take photographs such that in the one photograph you go only up to certain point, and in the second photograph start after that point. If you do that, the panorama function will never be able to put together the photograph. Every time there should be a little bit of overlap. Okay, and that is the same idea why we do what is called as partial digestion. Okay, so now once I do that partial digestion and put that in, then the beautiful thing is each of those partial things will now represent the entire genome. So all I do then is, if I take each of these clones and allow them to grow, take out the piece which is there, sequence it, and I put together the entire sequence, then I'll actually get the entire sequence of this genomic DNA. Okay? Is that clear?
Sir?
Yeah.
So how do we do this partial digestion? Because once the genomic DNA is experiencing all these 6 restriction enzymes, so they will be forming fragments which are non-overlapping.
No. Okay. So कैसा है मालूम है? Okay. I'll tell you how this is done. Okay. So if I take this piece of genomic DNA and I treat it with restriction enzyme, the ability to generate all of the 7 pieces depends on 2, the relative concentrations of genomic DNA and restriction sites. That is one factor. I should say restriction enzymes. And the second is the exposure time. Okay? That exposure time is very important. Okay? Because even though enzymes work fast, You can also always control how fast they work by altering the concentration that you're using, and second, altering, let us say, the temperature, the conditions in which, or the pH under which you are going to allow them to act. So what you essentially do if you want partial digestion is expose your genomic DNA to one, lower concentrations of restriction enzymes, and second, lower time. Okay, and since you have several copies of restriction enzymes, several copies of genomic DNA, the cuts will start occurring at random. But because you don't give enough time for every cut to be cut by every molecule of restriction enzyme, you end up getting what is called as a partial digest. Okay? Are you getting the feel how I
Yes,
do
sir.
it?
Yes, sir.
You are right in saying that if all sides will be cut, but all sides will be cut if I allow time for it. Okay. So typically what is done is you make sure the conditions are such that partial full digestion has not occurred. Okay. Okay.
Yes, sir.
Now, uh, now the second thing you can also do is use these clones that you have. Okay, what is called as a genomic library to locate where a certain gene is present. Okay, I'll give you a nice example. Okay, if I were to take the plasmid which is present, and what I mean plasmid is a recombinant plasmid which is present in each of these clones, and I take out that plasmid and cut it with the appropriate restriction enzyme. In this case, BAM-H1. Why BAM-H1? Because the way these clones were prepared was I took the genomic DNA, cut it with BAM-H1, then I took plasmid, I also cut it with BAM-H1, allowed these to come together, and I treated it with ligase. So this entire library here contains recombinant plasmids which are of this type, where this part is the same, which is the vector, and this part is different. And each of these collectively represent the entire genome. Now, if I have got a nice library and I want to take each individual clone, grow it, take out the plasmid and cut out the DNA and run that DNA on the gel, then each clone here, so here clone 1, clone 2, clone 3, clone 4, each is going to contain a different piece of DNA and they'll all migrate differently. Okay, see this blue line here? But then in each sample, I'll also get a constant band. Which is that constant band? That is this part. Okay, see this part? This is going to be the same in each and every plasmid. This is the one which is different in terms of length. So if I take each clone and I run it on a gel after cutting open the plasmid, I'll get two pieces, right? One piece will be constant, that is this vector part, while the other part which will be different is the insert part. Okay, and these inserts here collectively will actually represent the entire genome. Okay, now I can also do something very interesting. I can screen this for the presence of a given sequence by using a probe. Okay, and how would you do that? What would the technology be to do that?
RFLP.
No, remember the technology to detect something using a probe.
Acha, you can do X-ray type something.
Southern blotting. Remember
Southern blotting.
the southern blotting? So what I do
Yes, sir.
is I will take this information, put it on a nylon membrane, then I will denature this DNA, and then what I'll do is I'll make a suitable probe which will allow me to detect a certain gene and see which of these fellows carries that. Okay, and that is shown here. Okay, so I can do it in two ways. What is called as I can take the colony directly and open up the DNA then and check it. That is called as colony blotting. But I can do it more systematically. I can take that gel that I have, put a different probe and see where the probe is, what do you call as hybridizing and giving me a signal. So it is possible then that I might find that this clone here contains the gene of interest, and therefore I can get information about that gene by looking at this clone and sequencing that completely, because I might have used only a partial sequence. Okay. Are you getting a feel for how I will use the genomic library? One,
Yes, sir.
I can use this information to get the entire sequence if I don't know the sequence by sequencing each one individually. And second, what I can do is I can look for the presence of very specific pieces in this clone by using an appropriate probe to check for the presence of that sequence due to complementarity. Okay. Now, as opposed to this, we also have a cDNA library. Now, see what happens. If I'm doing a genomic library, though, they are suitable for cloning genes of prokaryote. And the reason is because prokaryotes generally tend to have very small genomes. Okay, even though we say 4 million, no, that is fairly, in a relative term, very small. So, typically, if you have 5,000 to 10,000 different clones obtained from partial digestion, they will represent the entire bacterium from which you are trying to, of which you are trying to make the library. Now, if you try to do the same with eukaryotic genomes, and remember, if you take an example of human, we have relative to 4 million, 3 billion, which is we are 1,000 times bigger than E. coli. Now, see what happens if you treat with restriction enzyme. So the number of clones you will end up getting will be 1,000 times this, no? So you'll get 5 million to 10 million clones. Now, to keep a track of 5 to 10 million clones is not easy. Okay. It's like saying, okay, remember that experiment that we did with Remington, and the Remington was 2,000 pages, and remember we had to make 10 copies of each page. So immediately our number of books were 22,000. Now imagine instead of Remington, you had another book whose size itself was instead of 2,000 pages, 2 million pages. To represent that, imagine how many notebooks you will need, each containing 1 page, 10 copies of it, and you will not have a library which will be big enough to house everything in a manageable fashion. Okay, so we never do what is called as genomic library of bigger organisms which have huge genes. Okay, and for those, what we do is we do cDNA library, because that now is more manageable. Because now what we say is, forget the entire genome. What I'm going to do is at least have a feel for, in this large organism, what is the scenario in a given tissue or a given cell. And sometimes that information is more important than just having information of the entire genome. Okay. So therefore, we have cDNA library. So these are what? These are collection of clones that contain copy DNA or complementary DNA that represent the genes that are being expressed in a given cell or a given tissue at the time at which you actually open the cell up. Okay. So, since the cDNA library is always representing the genes that are being expressed, why I say genes are being expressed? Because the starting material is going to be mRNA. And when do you get mRNA? When a gene is being transcribed, then only you get mRNA, which means the gene is being expressed. Okay. Now, this is very important. It represents a given cell or tissue at a particular period. What does that tell you? That I can have 2 cDNA libraries from the same tissue, but they could be entirely different depending on when I took the sample of the cell. Okay, as different as if I took it in the morning, afternoon or evening, or I took it when the animal was 1 year old, animal was 10 years old, animal was 50 years old, and so on and so forth. Okay, so it's a more dynamic collection, and you have to remember it will change depending on which genes are getting expressed at a given time. Okay. Now, they also will represent sequences after post-transcriptional modifications. Okay, that is splicing of introns. Because what you do is you do not use any mRNA, but you use what is called as mature mRNA. mRNA which has undergone all the processing after transcription. And if you remember, all those processing like introns are removed, and poly A tail is added, 5 cap is done. We use that mRNA to create the library. So in a sense, it is nice because it has a business part, that is the coding region of the genome. It does not represent all those sequences which have no coding function. So in a sense, cDNA library arguably, okay, at the moment is more to the point, okay, rather than a genomic library. So the issue is, how do we start making a cDNA library? Genomic library was very easy. All you have to do is take any cell, if you remember, and just break open the cell, break open the nucleus, and you'll get your DNA. And that DNA represents the genomic DNA. But how do I get mRNA? Okay, see the way they do it. Okay, what they do is they take beads, and these beads are typically made of saccharose or sephadex or any other inner material. And what they do is, to that bead attach a sequence of nucleotides which are all TT. Okay, so if I were to take a look at this piece of DNA in what you call as the 5' to 3' direction, it would, or I should say 3' to 5' direction, then it would contain all T's. Wherever you look, it will be T, 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 T connected to each other. Okay, now the reason I use these beads is because I know that all the mRNA in a cell which has undergone all the processing after transcription, that is removal of introns, addition of a 5' cap, and addition of a poly A tail, will all have this A tail. So if I make a bead which has got TT, what it will do is, if I expose that to all the mRNA content of the cell, it will bind to all the mRNAs because A and T is complementarity. Okay, so essentially what will happen when I put these two together, all those pieces of DNA or RNA which have a A tail will all be captured by the beads, and all those pieces which do not have A tail will simply go. Okay, then all I have to do is take these beads which are now holding on to all the mRNAs which have a poly A tail, and change the pH a little bit or change the ionic strength a little bit and release that mRNA. And now I have a whole collection of different mRNAs whose only commonality is that they have a poly A tail. Okay, so I have a collection of all the mRNA from the cell, and I obtain that by simply using what are called as beads which contain a T probe or a T complementary sequence or poly T complementary sequence. Now what I do after that? Now see what I do after that. I take that mRNA, okay, which I have separated from the beads, and to the solution of that mRNA, I add a poly T primer. So what will the poly T primer do? It will go and stick to this A, and I'll get a little hybrid here. Now what I do is add reverse transcriptase. And what does reverse transcriptase do? It reads RNA as a template, and to that it polymerizes DNA to give you a complementary DNA. But reverse transcriptase requires, in a sense, a primer to help it out. So this T primer provides that 3' OH end to which this reverse transcriptase can start adding DNA, and it generates this complementary DNA which is shown here in blue. So now after RT is done, I have a RNA DNA hybrid. Okay. Now what I can do is take this hybrid and degrade this mRNA either by using what is called as RNase H, or simply making the pH alkaline, because in alkaline conditions mRNA is very unstable, okay, and it simply breaks down. Now when that happens, what I get now is a DNA, and this DNA is actually exactly complementary to this RNA that I started with. Now when I leave DNA as is, it tends to kind of turn over itself. Now when it turns over itself, and if there are some sequences here which, let us say, fortunately are complementary to this end, I'll get a little hairpin. And when I get that hairpin, it is as if that I've got a primer here. So now when I add DNA polymerase, it presumes that this is a primer and there's a free 3' OH, and it starts adding the complementary DNA, complementary bases to the DNA which is present here, and I start making this sequence. And that will go all the way, and in the end I'll get A. Okay. Now see what has happened. I've actually made, by doing this, a DNA copy of the original RNA, which is represented by this strand here. Now, what I do is I treat with S1 nuclease. Okay. And what will S1 nuclease do? Where this DNA is single stranded, it will break it. And once it breaks it, I now have a double stranded, what is called as copy DNA. Now, all I have to do is take this piece, and to both ends here, I will add what are called as linkers, which contain sequences which are recognized by a particular restriction enzyme, and I will end up getting a whole bunch of different copy DNAs, all representing different RNAs, but now as double-stranded DNA, which have the same ends on both sides, at which there are restriction enzyme sites. So later on, when I take this sample and I treat it with restriction enzyme, I'll get this sequence, then a small piece here, which is what I've added, but a sticky end, and a small piece here, which I've added, plus a sticky end. And then I do the same thing to the vector, and when I put both of them together, I will generate a library. But now this library will only represent the mRNAs, not in their original form, but in their complementary DNA form. And such a library is referred to as a complementary DNA library. Okay. And now I can do the same thing as before. I can take that library, take out clones from it, they grow the plasmid, cut the plasmid out, look at the sequence, and try and identify which genes are represented by which clones. And then later on, I can do the sequencing and get the entire information of the sequence of the gene. But the difference is, I will get the sequence information which is not representing the genome, because remember, all the introns have been removed. So I'll get what is called as sequence information, but it will be of the final protein rather than the original gene. Okay. So what I'll get is that information which represents the protein that I obtained, because I'm using what is called as mature mRNA. But if I'm intelligent, what I can do is, from this information that I have of what is the sequence of complementary DNA, I can use suitable probes based on the sequence knowledge of this, what you call as a plasmid, which is hosting a certain complementary piece. Okay. To give you an example, let us say this is representing insulin gene. Okay, an insulin protein. So let us say then I take out this clone, and that is what represented that sequence. So if I were to take the clone from here, cut open the plasmid, and I sequence the mRNA, that mRNA sequence will represent the sequence of insulin. But indirectly, it is also going to represent the gene from which it was derived. So what I can do is, I can make probes from different parts of this copy DNA and use that to screen the genomic library if I have to, and then I can get which are the clones represent the sequence in the genomic library. And then if I look at the genomic library, I'll actually get the sequence of the original gene in its complete form, along with all the introns and exons. But that is not an easy job, but it can be done. Okay. So everyone comfortable with the concept of what is
Sir,
yeah.
sir, can you go to that slide where you have shown the strands and all?
Yes, sir. Yeah.
So, sir, at the end, you will be having only T and A in both the strands, right?
Yeah, see, here I will have TNA, correct? Here. Okay.
Yes.
So here will be T and here there will be A. Okay. So I don't worry about it. Okay. The reason I don't worry about it is, I know the main sequence is here, correct? This is where the actual protein ka sequence is. Okay. Now this is a poly A tail, and remember poly A tail is variable. Here I have shown 5. Sometimes it could be 10. Sometimes it could be 20. It could be 30. That doesn't matter. Even though that is there at the end, it doesn't bother me, because as soon as I do the sequencing, no, let us say I sequence this, I keep on going, and then I start seeing a lot of A's and T's. I know I have reached the point where there is a A or T tail. Okay. Also remember, somewhere here will be a sequence for a stop codon, correct? Because this mRNA is representing a protein, correct? So tomorrow when a ribosome sits on this mRNA and starts reading the codon all the way, it is not going to read poly A. This is never translated. This is just to tell you mRNA is mature and is ready for translation. In this mRNA, the stop codon will be somewhere here. Okay? And that will be represented in this clone. So I really don't worry about this DNA, even though they are there in all the clones. Does that answer your question? I think you are going there only, right? Okay, end
Yes,
same
sir. Yes, sir.
रहेगा.
हाँ, हाँ.
See, I don't care about it, because I know कि इधर किधर तो stop codon आ जाएगा उसका. और उसके बाद, see, I don't know, later on when we learn about transcription and translation, you realize that in a mature mRNA, some sequences here are not translated, and some sequences here are also not translated. The start codon is somewhere here, okay, and the stop codon is somewhere here. Okay. Now what do these things do here? These allow the ribosomes to come and recognize the mature mRNA, looking at the cap and everything, and then the ribosome starts to come here and searches for where is the start codon. And then when it starts to do the translation and makes a protein, it keeps on moving. Then it reaches a point which is called as the stop codon, and then it falls off. This part here is never read. That is called as the 3' untranslated region, and part of that is this poly A tail. So in this DNA, the bulk of the information, the real information, is contained in this part here. Okay, this represents, in a sense, the untranslated region and the untranslated plus poly A region. Okay, so you generally don't lose information about the protein by doing this technology. Okay, is
Yes,
that
sir.
clear?
Yes, sir.
Okay. Any other question?
Hello,
Anybody
sir.
has? Yeah.
Sir,
Okay.
about the hairpin formation.
Correct.
How does it happen?
Okay. See, this happens kind of at random. When you leave single-stranded DNA like that only, no, it starts to bend over itself. Okay. And there are random sequences present along the DNA. So if by chance, and invariably this occurs because all it needs is a few G's here and few C's here, or a combination of, say, let us say GCG and CGC. एक 3-4 चीज आ गया ना तो it will start bending over itself, because when it bends, the advantage the DNA gets is that all the in a sense non-polar bases are inside, and the backbone, which is sugar phosphate, is facing the outside, which is aqueous. So DNA, when you leave it alone, always tends to curl up on itself as and when possible, because it always tries to be more stable, because the environment is aqueous and the bases are hydrophobic. So this is just a chance event that occurs, and typically that occurs. Okay. Now if you feel that you do not want to wait for that, what lot of people will do is, after this DNA is made, no, they'll use another polymerase, remember, which can add a tail, and they'll add C's. So when you add a lot of C's, what will happen? I don't worry about the hairpin now. I have this 3' and a lot of C's here. Then what will I do next? Take a guess.
So there would be G's in the sequence, so the C's will.
No, what I do is I actually put a G primer now. I take a poly G primer and put it, and the poly G primer will end and stick here, and then I will do the polymerase activity. So you see what I'm saying? I can cheat. If I'm worried that hairpin nahi hoega na, I can take an enzyme which, remember, it adds a terminal basis without a template, and I put a string of C's, and then I make a poly T primer, a poly G primer, and the primer will sit here, and then I'll provide a 3' and I will translate. So now this cDNA will look very funny. Here I will have A and T, here I'll have C and G. But still, all my information is in the inside. Okay? Everyone?
Yes.
So you can cheat. People who do this are very sharp. सब सोचा है. जो भी तुमने question पूछा ना, अच्छा question है, and people have also thought about it. Okay? So they sometimes add a string of C's and then they use a poly T primer, poly G primer to do the this strand here, opposite strand. Okay? Everyone?
So,
Yeah.
sir, how do we add linkages at the ends?
We do what is called as blunt end ligation. Okay, remember I told you blunt end ligation is where you take a blunt piece of DNA which contains a certain restriction sequence, because here, if you remember, this is the 5' end, there is a phosphate here, there is a 3' end, there is a OH. Here there is a OH, here there is a phosphate. So if I take a piece of DNA which also has phosphate and hydroxy at the end, and it comes here and it just sits here, ligase can potentially join it together. Okay, and this end and this end. So what is called as polymer tailing or blunt end ligation. Okay, this takes a little time, but it happens easily. So I just make a piece of DNA of the sequence that I want, make sure the ends are blunt. I let those blunt ends come here and seal it with a ligase. So ligase will now seal 2 ends here, and I'll get a foreign piece. Okay?
Yes, sir. Okay.
Any other question?
Sir, can you explain the next slide again?
Which next slide? This one?
The one after this. Yes.
See, this is the same thing I told you last time. So what I can do is, I will have now a collection, no? I'll have, when I do this experiment, I'll get a lot of pieces, each containing a copy which is slightly different, which is representing a different mRNA which is obtained in the tissue. Okay. So remember now, when I take a liver cell and the liver cell is making 10, 20 different proteins, it will have 10 to 20 different mRNAs. This process will occur with each of those mRNAs, and each will end up giving me a piece which will be a variable length and variable sequence, but whose ends will be almost the same. And now I take this entire thing and I put it back into a bacterium, right? By what is the cut and paste technology. So I'll end up getting clones. Now these clones don't represent the genome, but they represent all the different mRNAs from which I made copy DNA. So for example, now I want to find out, let us say among this, which of those clones carries the gene for, let us say, quote unquote, insulin. Okay, presume this was from pancreas. So I know the sequence of insulin. Based on that, I will make a probe, because I also know nowadays, okay, what code codes for which amino acid. For example, we know AUG is coding for methionine. Like that, I know CUC codes for, say, phenylalanine. So if I know insulin ka sequence, I can use that sequence and figure out what would be the RNA sequence which I need to give me this protein. And I use that to make a probe. And I use that probe and hybridize it, and I find, okay, among this, which of those clones is carrying the gene for insulin. Okay, that is what this is saying. So this is just showing kind of southern blotting in a sense. Okay, same as I did for genomic. I just repeated that slide here. Okay, although upper it says no screening genomic library, it should say screening cDNA library. Now, what some people do, and look at the statement here, use of antibodies. If I am intelligent, no, what I will also do is instead of putting all of these sequences in plasmids, which will just replicate the plasmid, I can put it in what is called as an expression vector. An expression vector is where not only will the plasmids be duplicated, but also the sequences which are present on the plasmid will be expressed. So what will happen now? All of these different clones will all express different proteins which are represented by the mRNA they are carrying in the plasmid. So then what I can do is, let us say this one is producing insulin, because that plasmid contains the sequence for insulin. I can take an antibody to insulin and I throw it in there, and it will bind here, and we'll be calling that Western blotting. Okay, so I can do detection of those clones which are producing specific proteins by making antibodies to the protein which I want to have information on. Okay, does that make sense? Hello?
Yes.
Yes, sir.
Yeah. Okay, so I'm going to stop sharing here. I'll check to just a second. Let me check if there is anything in the chat box. Sometimes, what is the use of junk DNA? Okay, let me see. Starting. As I say, because it is called junk DNA, is probably why. Probably because maybe it doesn't have any use. Okay, but it is more correctly speaking, we don't know what its use is. Okay, and it cannot be junk because 99% of our DNA is actually junk. Okay, only 1% codes for genes. Okay, so if only 1% is coding for the gene and 99% is apparently there for no reason, over evolution we would have lost it. No, why would we keep carrying that baggage with us all the everything all the time? Okay, so which I feel is telling you that there is probably some role for this DNA, which for the moment we don't know. Okay, and as we realize a little later what their uses, they will not be called as junk DNA. Aditi, she's asking, isn't there any way to get all the coding sequences of the whole DNA in one go? For now, we rely on cell signals which generate different mRNAs in different conditions, and then we harvest the mRNA. So for different sequences, we have to take new cells every time, and it becomes tedious to do. So isn't there a way around it? Yeah, there is potentially a way around it, and potentially way is a computational approach. Okay. Now, what this computational approach relies on the fact that irrespective of which protein we are going to talk about, there are some aspects of the protein which the information, especially in the untranslated region, which are common for a given organism. For example, in humans, doesn't matter which protein I make, I'm using the same ribosomes, right? What we call as the ATS ribosome. So I should have a way that these ATS ribosomes recognize all the mRNAs which they have to do translation on. So there needs to be on the mRNA something which is common to every protein that is going to be made later on. And that common sequence will always be present not in the real translated region, but in a region which is not there for translation but for recognition by the ribosome. So, and since those sequences are fairly constant, what I can potentially do is search for these sequences in my whole genome. Okay, and I can say that wherever this genome, this sequence appears, it is probably representing a gene which is going to be later made into a mRNA, because these sequences are very important for recognition of by ribosomes of how to get the translation going. Okay, that is great. Okay, so that will tell you that. But what happens is, after that, when you get information about the sequence, no, you will get the entire sequence information with all the introns information also in there, and that is a little difficult to delineate in terms of out of that whole sequence what actually represents the final protein. Now people are getting good at that also, recognizing where the cut, introns start and ends are there, which also seem to have some commonality. So nowadays people use actually computational techniques to what is called as mine your genome for this information. Okay, so there is a way around it. It is a computational way, but it is still not a foolproof way. But we are getting good at it. Okay, so does that answer your question, Aditi? There is a way around it, but it's based on knowing a lot of things about how our mRNAs are organized. So we use computational technology to do that. Okay. Before addition of probe to DNA, we need to make it single stranded. Yeah, always. The probe will only bind when you take your DNA and separate it so that it becomes single stranded. Otherwise, it will not. Okay. So I think I have answered all the questions. Aditi, did you get the answer to the question you are asking? Maybe you can put it in the chat box, yes, no, or just unmute yourself and say yes, no. Shivam, I answered your question also. Okay.
Yes, sir.
Okay. Oh, maybe Aditi has already. Okay, yes, sir. I got it. Aditi said. Okay. So can I stop recording? Okay. I'll stop recording and then I'll